Great. Thanks very much, Gloria, for the introduction, and thanks to the Research Office for organizing this event. So I'm just going to start out here by giving an overview of the Center for Systems Neuroscience. Gloria already mentioned this. The center includes a broad variety of faculty from all the different schools, colleges, and departments at Boston University. I've laid out some of the goals of the center as they were proposed in the charter for the center just over 5 years ago. One of these goals is to foster collaboration between neuroscience researchers here at Boston University. This Research on Tap event is exactly that type of event to bring people together. So thanks to everybody for participating. We have people here from the School of Medicine, from the Sargent College, from the College of Engineering, from the College of Arts and Sciences. So it's great to see the diversity of neuroscience research here at Boston University. Another goal is to enhance the profile of neuroscience research at Boston University. For this, we've been having these many seminars with the great luncheons afterwards. We're having a workshop in May on the 20th and 21st that Ben Scott and Chandramouli Chandrasekaran have helped organize, and that the notices about that have been going out. We have just established a Twitter account for CSN. View CSN Neuro, so please send us news that we can tweet about. If you have a publication or award or something of that sort, to get the name out there. Another goal has been to attract talented young investigators to Boston University. I think we've been very successful in that. We've had a lot of marvelous young junior faculty join in neuroscience over the past few years. Also postdoctoral fellows and students. The faculty have received a number of awards and grants, successful grant applications. We're here to help support cutting edge research in neural systems. One of the things that we're doing for that is to support Center for Systems Neuroscience fellows that are postdoctoral fellows that are coming to Boston University bringing new techniques to kind of foster further development. And finally, as Gloria mentioned, we want to encourage strategic planning about future faculty hiring in some of the initiatives that we've had for across the campus hiring and the new data science initiative that will be coming up, as well as the development of collaborative funding applications. And I encourage you to get involved. We've had a retreat on October 30th and talked about these various strategic initiatives, and I encourage you to continue to be involved in that. And then I want to thank Gloria Waters for providing all of the funding for the Center for Systems Neuroscience, and again, thank the Research Office for organizing this Research on Tap event. All right, so now the timer is going to start for me, just like it will for everybody, so I've got to do this in 4 minutes. I don't know when is it Brian that's doing the timing? So that's the guy to watch. All right, and I've timed this, and it comes under 4 minutes, but only just barely. So here we go. So in my laboratory, we do work on coding of space and time in the cortex. This includes work using both, oops, I hit the wrong button there, both electrophysiological recording and imaging in the hippocampus, the entorhinal cortex, and the retrosplenial cortex. And we're interested in general principles of cortical coding for episodic memory and goal-directed navigation. We have collaborative support from Office of Naval Research MURI grants, including work with Yanis Paskalidis related to robotic navigation. Now, probably many of you have heard of grid cells. These are neurons in the entorhinal cortex that code location. Here's an example of a grid cell. The gray line shows the trajectory of a rat as it's running around foraging for bits of crushed up cereal in a 1 meter square environment. And the red dot shows the location of the rat each time a single grid cell fires a spike, right? You can see it's very stably firing when the rat is in specific spatial locations. Now, one of our major findings was that the cells that fire as grid cells in the environment will also fire as time cells. So the rat, after doing this, running in the environment, the rat's running on a treadmill in one location in one direction for 16 seconds, then it does a left turn, then it runs for 16 seconds, and it does a right turn. And on each of these trials, this same cell is firing at specific time points, at the beginning, at the middle, and the end of that 16-second period. So the same cell that fires based on spatial location is also firing at different time points as it's in a single location. And this could be important for episodic memory that's defined by events occurring at particular locations and times. Now, in a different study, Will Mao did endoscope one-photon calcium imaging of these time cells and could record a large number of cells. You can see these cells are tiling the 10-second delay period here in this task run by a mouse. And it's actually changing over days, which would be important for being able to code episodic memories on different days. Now, we've collaborated with Mark Howard in modeling this distribution of the time cell responses. Now, in further work, we've been interested in the mechanism of generating these spatial responses of neurons. One of the things that has been kind of a standard model of grid cells is to do integration of the velocity of the animal. And that's consistent with the fact that many cells in the entorhinal cortex will change their firing rate with the running speed. But as shown here, this is happening over long time periods, but when we look over shorter time periods, it actually can often show a lack of this firing rate coding or the opposite direction of firing rate coding. And that's a potential problem for these path integration models of the spatial representation. So in exciting recent research, we've been exploring an alternate idea that the spatial coding in allocentric coordinates could arise from egocentric coding of the boundaries of the environment. This is work done by Andy Alexander and others in the laboratory. Great. So Andy Alexander is recorded in retrosplenial cortex and has seen neurons that have this egocentric representation. So like I know my position in this room by knowing where the walls are in my spatial view. So here you can see in the same foraging task, there's a neuron that's responding when the rat's running near the north wall going west, or near the west wall going south, or no, the south wall going east. And if we look at this, you can think of these spikes in egocentric coordinates. For this one spike, we can see, oh, here the rat is facing this direction. We can plot the boundary relative to the rat, the front, back, right, left of the rat. And then we can sum up for all the different spikes, where was the boundary? Well, on average, it was towards the front right of the animal. Here's another example where the boundary was towards the left of the animal or towards the right back of the animal. So this is evidence that there are egocentric representations of boundaries that could form the basis of forming this allocentric representation of spatial location in the entorhinal cortex. And I'll conclude there.